Good morning, everyone, and welcome to Sensogen's GARD technology webinar. This webinar focuses on the principles of the GARD technology platform and will give you in-depth knowledge about the GARD assays for skin and respiratory sensitization testing. Today's presenter is Angelica Johansson of Sensogen. She's a research scientist and technical support manager and have more than 5 years experiences from GARD and the technology. And with us during the Q&A session is also Dr. Henrik Johansson. Henrik is Sensogen's chief scientist and has a PhD in immunotechnology. He is specialized in vitro assays for predictive immunotoxicology and is a co-developer of the GARD technology platform. Again, welcome everyone. I will now give the word to Angelica Johansson. Angelica, please go ahead.
Thank you very much, Tina, for the introduction. And I would also like to take the opportunity to welcome everyone to today's webinar. I'm very glad to see that so many are interested in our technology. The focus is to give you a brief introduction to Sensogen and to the GARD assay, and I will try to describe how we can use a combination of genomics and machine learning to create next generations of in vitro testing platform for assessment of chemical sensitizers. Before I go into the specific detail, I would like to give you a brief introduction to our company. Sensogen was founded in 2010 as a spin-out from the Lund University, and today we comprise a highly multidisciplinary team of 20 employees. We have our headquarters in Lund in Sweden, and we have a sales office in the US. Since 2017, the company has been listed on the Nasdaq First North stock market. Our lead product today is GARD, which stands for Genomic Allergen Rapid Detection. It is a state-of-the-art testing platform for assessment of chemical sensitizers. Today, the platform has 4 readily available assays: the GARD skin assay, the GARD potency assay, which can be used for hazard assessment and potency assessment of skin sensitizing chemicals, the GARD skin medical device assay, which can be used for skin sensitization assessment of medical devices, and we have the GARD air assay that can be used for identification of respiratory sensitizing chemicals. Sensogen is continuously working to develop new assays, as well as to improve and extend our available assays. Our business model is to sell direct and via partners to make the GARD assay available globally. Our vision is to replace animal experiment and to establish a new industry standard. Working within the field of predictive toxicology, the challenge for us is to translate the complex biological endpoint into a tool that can be used for safety assessment. Ideally, we want our assays to provide biologically relevant results with a high predictive accuracy. We want them to be simple to perform and give the current paradigm shift to move away from animal testing. We, of course, want our assays to be performed in vitro. So traditionally, there has existed at least a conceptual trade-off between the simplicity of a model and the informational content it generates, on which we can base our decisions. While animal methods often provide high informational content, the majority of the available in vitro assays, although being quite simple to perform, is based on few biomarkers and provide very little mechanistic information. Our ambition with the GARD assay is to eliminate this trade-off and develop in vitro assays with a simple workflow that provides sufficient information for the end user to trust the results. The problem with choosing a too simple model, after relying on only 1 or 2 biomarkers, is that they normally fail to represent the real complexity behind the biological endpoint, preventing you from seeing the full picture. However, by adding parameters to the readout and increase the mechanistic information, you can start to build a view that better represents this underlying complexity. And in the end, of course, you would like to see the big picture. The same phenomenon can also be illustrated in perhaps a more relevant context by looking at the coordinate system where the X and Y axis correspond to measurements of 2 relevant biomarkers. Obviously, these 2 biomarkers do not contain sufficient information to distinguish between all the sensitizers and non-sensitizers. However, if you add a 3rd relevant biomarker to your readout along the Z axis, you will be able to better capture the complexity involved in the sensitization reaction and to distinguish the sensitizers from the non-sensitizers. This transfer from few data points toward a biomarker signature with higher informational content forms the basis of the GARD strategy. What I really would like to emphasize in this presentation is that the GARD assay is not actually one single assay, but it's rather a testing platform on which you can base several different toxicological endpoints. I will now talk more about the GARD technology platform, how the assay was developed, and how human relevant cells in combination with genomics and machine learning creates the base of our in vitro assay. All the applications of the GARD platform share a similar structure, comprising, firstly, a relevant biological system, secondly, a training dataset of reference substances for data-driven genomic biomarker identification, and thirdly, a predictive genomic biomarker signature identified from genome-wide profiling, and lastly, a prediction model based on machine learning and pattern recognition algorithms. To describe how we develop our in vitro assays in a general view and not focusing on a specific application, I would like to go through the different modules in detail. The first and perhaps the most important step for construction of a mechanistically relevant in vitro assay is the selection of an appropriate cell system or biological model capable of reflecting the adverse toxicological endpoint that we are trying to predict. To describe this, we can use the adverse outcome pathway for skin sensitization as a point of departure. The AOP is basically a way of structuring the information to describe the key event, starting from a molecularly initiating event of hapten formation to the final adverse outcome of allergic contact dermatitis. For respiratory sensitization, there is still a large knowledge gap related to exact mechanism by these chemicals give rise to occupational asthma. Dr. Ian Kimber described in a publication in 2018 that the key events for the development of skin and respiratory sensitization are actually pretty much the same. The differences are of fine details. One of the main mechanistic differences between skin and respiratory sensitization is represented by generation of various T cell populations, where Th1 response associated with skin sensitization and a Th2 response is associated with development of respiratory sensitization. The dendritic cell is a highly relevant cellular model, since they are responsible for driving the polarization of naive T cells into either Th1 response or Th2 response, which again represents the key mechanistic difference between skin and respiratory sensitizers. Regarding a cellular model for respiratory sensitization, it is also important to point out that sensitization of the respiratory tract may be acquired also through skin exposure. This means that a relevant model does not have to be based on inhalation exposure or on cells specific only to the respiratory tract. As a cellular model in GARD, we use a human dendritic-like cell line called sensor cells. This is a monocytic cell line that has been differentiated to acquire characteristics that are identical to those of primary cells. Similar to primary cells, sensor cells are capable of taking up and presenting antigen on MHC class 1 and 2, as well as CD1D. They respond to pro-inflammatory cytokines by upregulation of co-stimulatory molecules such as CD80 and CD86. And importantly, they contain the functional elements involved in regulation of Th1 and Th2 polarization. Having defined the cellular system, the question is then how we monitor the reaction of these cells to skin and respiratory sensitizers. We know that these cells react in a multitude of different ways upon encounter of antigens. Firstly, they are equipped with various receptors that recognize foreign substances. These receptors may in turn activate various self-defense mechanisms of the host. You might have cellular stress responses such as oxidative stress. And finally, they express receptors that allow them to migrate and communicate with other cells of the immune system. The question is then how we best monitor all these events at the same time. All of these changes are carried out by proteins, which in turn is controlled by the transcription of DNA. So we believe that genomics, or more specifically gene expression analysis, is a very efficient way to get a complete view of what is going on in a cell upon chemical stimulation. With this knowledge and assumption, we early on hypothesized that there should exist genes in these cells that were differentially regulated depending on the stimuli. And if we were able to find such genes, they could be used as predictive tools. And this leads me to the second module, the definition of a training dataset. A training dataset comprises a set of well-characterized chemical compounds with known expected outcome. This means that the chemicals should be annotated according to their sensitizing capacity, which ideally should be based on human experience. Moreover, the training data should cover a large chemical space, both with respect to chemical reactivity domains and different industrial applications. This will enable for capturing different modes of actions and to develop models with a large applicability domain. The distribution in these plots in this slide shows the datasets used to develop our potency model, which I will describe in more details later in this presentation. To examine which genes that are differentially activated in the sensor cells, the compounds in the training datasets is used for cellular stimulations. After incubation, we extract RNA and measure the complete transcriptomes of the cells using the Affymetrix microarray platform. So for each chemical in the training set, we measured 29,000 genes. And this leads us to the next module in our platform, which is the identification of a genomic biomarker signature based on how the compounds in the training datasets interact with our biological system. And starting from the 29,000 genes, and by using a completely data-driven approach, we then identify a subset of genes that are differentially regulated depending on whether cells were stimulated with a sensitizer or a non-sensitizer. So in contrast to the traditional approach, we won't often measure a single biomarker based on previous experience. One of the largest advantages associated with this data-driven approach is the possibility to actually see how the cells react to the different chemicals and to identify novel and more predictive biomarkers for sensitization. And later in this presentation, we will see how this results in both a high predictive performance as well as in a large applicability domain. Once we have identified our biomarker signature, our ambition is to transfer our gene expression measurement from measuring the complete transcriptome of the cells to a higher throughput gene expression platform that measures only the genes in our signature. The Affymetrix platform that we use in order to identify our biomarker signature is extremely useful, as you can look at the whole transcriptome of the cells and you can focus on those genes that are highly predictive for the assay. However, the platform does not fit very well into routine testing because it's very label intensive and it's very hard to automate with low sample throughput. Therefore, what we do after we found a biomarker signature is that we transfer the genes to another hybridization-based platform that is called NanoString. The benefits here are, of course, because we can do the analysis in 24 hours with only 15 minutes hands-on time, and we can simultaneously increase the sample throughput to 384 samples per day. What you see in this picture is a comparison between the Affymetrix microarray data and the novel NanoString data. As you can see, the gene expression measurements could be readily reproduced and validated on the NanoString platform, demonstrating that the genomic biomarker signature is a stable and fixed biological endpoint, independent of its technological means used for gene expression measurements. All our GARD assays are based on the same technology platform, using the same biological system, but the difference between assays are the available training data and identified genomic biomarker signature. And as you can see, the signature consists of 200 genes for GARD skin, 51 genes for GARD potency, 28 genes for GARD air, and GARD skin medical device comprises the same 200 genes as for GARD skin. After having identified and transferred a predictive biomarker signature to NanoString, we now need to define a prediction model that contains the basic rules on how to use gene expression data in order to classify unknown substances. To assist us in this process, we use a modern technology for pattern recognition, which is often referred to as machine learning. In the same way as people learn from their past experiences, you can also train a computer algorithm to learn from past experience. And this is basically the definition of machine learning. I would like to describe how machine learning works and how we use this very powerful technology in the GARD assay. The machine learning algorithm learns by observing a set of objects with known expected outcome. If you consider our training data, this is actually exactly what we have. We have the gene expression profile from a set of well-characterized compounds that are labeled as sensitizers and non-sensitizers. Using this training data as input, the machine learning algorithm learns how to distinguish expression profiles induced by sensitizers and non-sensitizers. For classification of an unknown sample, the expression profile induced by the test substance is compared to the expression profile of the training samples and classifies either as a sensitizer or a non-sensitizer. In this example, the expression profile of the test substance was most similar to the expression profiles of the non-sensitizer and was therefore classified as a non-sensitizer. This is the graphical representation, but we can of course also do a more mathematical. The information in the heat map can then be expressed as a gene expression data matrix containing information of the class label. In the machine learning algorithm, this now becomes an optimization problem. What the machine does here is to assign different weights to the different genes with the purpose to optimize the discrimination between the sensitizer and the non-sensitizers. This will lead to a fixed mathematical formula that never changes. Every time that you try to feed in some new data in your test data, you will have exactly the same basic mathematical structure. The gene expression data from the test compound, now without the label, can then be inserted into the fixed formula and the model provide a label to the test compound. There are today many different machine learning strategies available in the scientific literature, and the specific machine learning algorithm that we use is called support vector machines. Briefly, I would like to exemplify how the SVM work for a model based on 3 biomarkers. You have your training data plotted in this figure based on gene expression of the 3 biomarkers. So what the SVM does is that they find a hyperplane that best separates the sensitizers and the non-sensitizers in the training data. And the prediction of the unknown sample is based on the decision value, which is basically only the distance between the unknown testing compound and the separating hyperplane, where a positive value is classified as a sensitizer and a negative value is classified as a non-sensitizer. In this example, there's only 3 biomarkers, but the strategy and interpretation are the same for higher number of biomarkers. As for GARD skin, comprising 200 biomarkers, the plot to the right is showing some real data from a GARD skin experiment. Having defined all the different modules of the GARD platform, I would now like to continue the next part of this webinar and explain how we use the finished model to provide hazard assessment of unknown substances in only 6 steps. So in practice, what we do today is that we stimulate our in vitro models on the Dietrich cell with any chemical substance that is to be tested. The cells are stimulated at one single concentration, which we call the GARD input concentration. To determine this concentration, the first thing that we need to do is to screen for cytotoxic effects, aiming for a concentration that induces a relative viability of approximately 90%. If a compound is not toxic, we use either the highest soluble concentration or a default concentration of 500 micromolar. Once we have established the GARD input concentration, we perform cellular stimulations again. At this step, we also perform a quality control with cells by measuring common DC maturity markers using flow cytometry. After 24 hours of incubation, we isolate our genetic material. We control the RNA quality and measure the gene expression of the genes in the biomarker signature using the nanostring platform. The gene expression profile induced by the unknown testing compound is compared to the expression profiles induced by the well-characterized compounds in the training dataset. And final classification are then provided instantly and transparently by the SVM prediction model. Now I would like to give you a bit more information about the specific assay that we have available. And I would like to start with the GARD skin assay, because this is the first assay that we have developed, and this is also the model that we have the most data available from. The GARD skin assay was developed in the same way as described earlier. We used a training dataset comprising 20 sensitizers and 20 non-sensitizers, balanced in terms of chemical, reactivity domain, and industrial applications. We stimulated the sensor cells and measured the complete transcriptomes of the cells. And we could identify 200 genes that were differentially regulated depending whether the cells were stimulated with a sensitizer or a non-sensitizer. And we collectively refer to these genes as the GARD prediction signature. I guess that one of the probable explanations to why GARD performs well is because it does not only measure one specific endpoint, but we measure a whole set of different endpoints that are immunologically relevant to the sensitization reaction. When we actually look at the specific genes that we have in our prediction signature, we can see that they don't only have one single mode of action, but they are rather involved in a broad spectrum of different pathways that are involved in the development of skin sensitization. If we try to map these genes in our biomarker signature in the different key events in the adverse outcome pathway, they cover a broad spectrum in the different key events that are represented in the AOP of skin sensitization. Transparency is very important for us. We always publish our data in order to enable full insight into the GARD assay. We have also published all assay development steps for GARD skin, as well as the complete list of genes for this assay in peer-reviewed scientific journals. I would now like to move over to the next assay that we have available, which is our GARD potency signature. The GARD potency signature is capable of subclassifying chemicals according to the CLP classes, which is strong sensitizers and weak sensitizers. You might start to recognize the strategy that we use. We use the same biological system. On top of that, we use the training data comprising 22 strong sensitizers and 29 weak sensitizers. And we use this information in order to extract genomic biomarker signature. In this slide, I show you that the GARD potency is comprised of 51 genes. So on top of that, we also, of course, add our prediction model. As you can see in the heat map, we can actually see that there are many genes that are differentially regulated depending on the inherent sensitizing properties of the chemical. The intended use of this signature will be in a tiered approach, where sensitizers are first identified in GARD skin and then further subclassified into strong and weak sensitizers in GARD potency. GARD skin and GARD potency is now progressing towards regulatory acceptance through a recently completed ECVAM validation study performed in cooperation with 2 external laboratories, Burleson Research Technologies in the US and Eurofins in Germany. The validation study involved a ring trial where a total of 28 compounds were classified in a blinded fashion in 3 consecutive experiments in each of the 3 laboratories. This is the data from the validation study of GARD skin. The chemicals to the left of the dotted line, they are the true sensitizers, and the compounds to the right is the true non-sensitizers. The positive values were classified as a sensitizer, and the negative value were classified as a non-sensitizer. And as you can see from this figure, there's very little variance between the different experiments, as well as between the different labs. Most of the sensitizers and the non-sensitizers were correctly predicted. And if we put this in numbers, we experienced a between laboratory reproducibility of 92% and an accuracy of 94%. It is very well balanced between the sensitivity of the assay and the specificity of the assay, which is quite unique for the current in vitro tests that are available. We further subclassified those chemicals in the validation study that was classified as sensitizers in the GARD skin into 1A and 1B using the GARD potency prediction model. And this data has also been submitted to regulatory authorities. So for the binary prediction of sensitizers into 1A and 1B, the ring trials show an overall accuracy of 82%. The validation data for both GARD skin and GARD potency have been submitted to ECVAM and OECD for scientific review. In the meantime, we do have a formal statement from ECHA that test results from the GARD assay can be used for REACH registrations to inform on key event 3 in the AUP for skin sensitization. The third assay I would like to show you today is the GARD air assay for identification of respiratory sensitizing compounds. We were fortunate enough to receive external funding from the EU Horizon 2020 project for the development of the GARD assay and eventually bring it on into industrial application. And I would like you to show the results and how we have developed this assay. We used the same approach as described earlier. We stimulated our cells with a training data set comprising 10 respiratory sensitizers and 20 non-respiratory sensitizers. And again, starting from measurements of the complete transcriptomes of the cells and using a completely data-driven approach, we were able to identify a set of 28 genes which were differentially regulated depending if the cells were stimulated with a respiratory sensitizer or a non-sensitizer. And by looking at the identity of these genes, we could again confirm that they were immunologically relevant and can be mapped to the AUP for respiratory sensitization. Interestingly, in the signature, we found several genes involved in innate immune activation processes, as well as genes involved in the polarization of T cells towards the Th2 process, with the most well-characterized genes being involved in the TSLP receptor signaling. So we went further to evaluate the performance of this signature on a blinded external test set comprising 20 chemicals. And on this specific dataset, assay was capable of correctly predicting 90% of the compounds with a sensitivity of 100%, which means that all respiratory sensitizers were correctly predicted by the assay. Importantly, except for one single test substance, all skin sensitizers tested in the assay this far have been correctly predicted as non-respiratory sensitizers, indicating that this model were also capable of discriminating respiratory sensitizing chemicals from skin sensitizers. We are currently in the process of summarizing the results from a blinded ring trial study where we have evaluated the GARD assay even further. The last assay that I would like to show you today is the GARD medical device assay for identification of skin sensitizing medical devices. So this assay development was performed a little bit different. First, we identified the criteria or requirements of the method. We needed a polar and non-polar extraction vehicle that was compatible with the GARD assay and also described in the ISO standard. It also needed to be sensitive enough to detect low levels of sensitizers leaking out from material into the extract. And we started to finding oils that we can use in the GARD skin assay. We optimized the GARD skin protocol, and we used the optimized protocol to demonstrate that GARD skin can be used to assess materials spiked with known skin sensitizers. And this is what we used in the in-house validation study. It was performed using saline and 2 different oils. We assessed silicone and TPO use spiked with 5 known skin sensitizers. We also analyzed known sensitizing materials, and we had relevant controls and used extraction conditions found in the ISO standards. And the result from this in-house validation was very clear. All material was assessed correctly, and we could happily make this assay available. So when I've now explained the basics of GARD, machine learning, and genomics, I would like to talk a little bit more about the features and benefits of the GARD platform. As been explained earlier, we have sent in the ring trial results and documents relevant and are included in the OECD test guideline program. ECVAM and OECD identified concerns regarding IP and GLP for cloud-based software, and SensaGen have worked actively with OECD to get the new guidelines concerning IP and GLP compliance in place, and is confident that the requirements will be fulfilled. We are now waiting for the expert group to start reviewing the assay during this year. As explained earlier, GARD is accepted by ECHA for REACH dossier, and GARD potency makes it possible to classify the test chemical into CLP classification of 1A and 1B. And currently, the SensaGen headquartered lab in Sweden are working towards getting GARD fully GLP compliant and expect audit and approval in the spring 2020. Another popular topic when evaluating the performance of different in vitro assays is, of course, their applicability domain. This slide is showing the number of chemicals that we have tested this far with respect to the different industrial applications. GARD has been used for testing several groups of traditional difficult-to-test samples, such as pre-prohaptens, low water-soluble substances, surfactants, and various complex mixtures, such as UVCBs, natural extract, e-liquids, and finished products. We consider that GARD skin is applicable for a wide range of chemical ingredients used in sections such as industrial chemicals, agrochemicals, cosmetics, pharmaceuticals, and household products. The plot to the right shows bars where the numbers are of chemicals in each industrial sector, and the dots on the top are basically how the accuracy varies for the different industrial groups with respect to the mean accuracy of the assay. So we have a mean accuracy of 89%, and as you can see, the accuracy is not very far away from this measure for any of the groups that we have tested. This indicates that we can test many different industrial applications. One thing that has been suggested is the pre-prohaptens would be really hard to predict in vitro systems because they need some form of activation in order to create the allergens. So if you look what genes are actually present in our dendritic-like cell line, we do have a lot of metabolizing enzymes that are actually capable of converting prohaptens. Therefore, in theory, it should not be any problem for the assay to test pre-prohaptens. The other thing that some often say is that it's difficult to test substances with high optimal partition coefficient or those with other properties that will result in very low water solubility in aqueous media. What you can see in this slide is that pre and pro-hapten is in general not an issue for dendritic cell-based assays. And if we look at those compounds with high log p-value, we see it seems to work quite well also in the GARD assay. Pre and pro-haptens and compounds with high log p-values has been one of the main arguments to incorporate 3D-based cell models, but I think this slide shows we can actually test them in the available assays that we have already. The complex mixture are the biggest challenge as they have an uncertain composition, often with solubility issues. As we advance the GARD platform, we have learned more about its potential to use additional solvent, which is an important factor to succeed with testing of complex mixtures. Today, we have tested several types of complex mixtures successfully, such as natural extraction or agrochemical formulations. This slide is showing an example of the importance of extending the solvent selection to get the test substance in solution. The first and second plot is showing the test substance not soluble in DMSO or water, and then when the test substance has been truly solved in DMFO glycerol, it can be predicted as a sensitizer. All the GARD assays can be used individually, but they can also be used in a combination. The advantage with this is that it is possible to simultaneously determine if a given test substance is a respiratory sensitizer, a skin sensitizer, or a true non-sensitizer. And of course, if the substance is classified as a skin sensitizer, it is also possible to characterize the specific hazard by adding the readout from GARD potency. We consider that GARD is very efficient. It's a quick and gives you reliable results. It has a short test time of 2 weeks. It's less expensive than animal testing, and we give you the opportunity to pre-test difficult to test samples. So how do I, if I would like to test my substance in the GARD assay? Of course, the first thing that you need to do is contact us, and we'll be really happy to help you design an appropriate testing strategy. We have great experience in the study workflow and always try to keep the administrative parts as short as possible to facilitate the laboratory personnel to enable the test time to be as short as 2 weeks. Today, we have several CRO partners that are up and running globally. In the US, we have collaboration with Burleson Research Technologies and MB Research Laboratories. And in Europe, we have a CRO partner in Eurofins in Munich. And of course, in addition to that, we have a global map of the distributors that can help you organize your testing. Now, by that, I would like to round up this first webinar, and this was basically a general introduction to GARD and to our assays. Feel free to contact any of our key account manager, Andrew Forward in Europe, or Joshua Smith in the US to get more information if you're interested in testing. Thank you so much for listening today.